Hello everyone. In this presentation, I am going to explain the general characters of Archaebacteria. These were the bacteria once considered to be the extremophilic organisms, that means which can live in the extreme environmental conditions like high temperatures and in the acidic environments, and even they can do unusual things like production of methane, and the organisms are more diverse ecologically and physiologically. In this presentation, the contents include the introduction, morphology and cell structure, physiology and metabolism, and classification of this archaea. The term archaea is derived from a Greek word archaeos, that means primitive or ancient. They possess the primitive structure and the ancient energy metabolism. Some of the first Archaebacteria were discovered in the Yellowstone National Park's hot springs. The habitat includes, they are found in the extreme aquatic terrestrial habits like deep sea vents, saline waters, hot springs, and even below petroleum deposits. They are mostly anaerobic and they live in the low oxygen environments, though they are oxic, but they live in the low oxygen environments. Many of the Archaebacteria cannot be cultured in the laboratories. The organisms in this domain, they share the characteristics of both bacteria and eukaryotes. They possess the distinct characteristics that make them into a separate domain, archaea. Moving to the morphology, the size of the archaea ranges from 0.1 to 15 micrometers, and they exist in different shapes like spirals, rods, plates and spheres. Even some may appear even in the flat or square shaped also. They are irregularly shaped and even pleomorphism, they show the pleomorphism. Even some exist in the form of a single cells and some others form like filaments or the aggregates. If they are present in the form of filaments, they can grow up to 200 micrometers in length. Unlike to that of bacteria, the cell wall of archaea is made up of a pseudopeptidoglycan. It is not a true murine layer, means the true murine layer contains the alternating units of N-acetyl muramic acid and N-acetyl glucosamine, but here the bacteria possess N-acetyl talosaminouronic acid instead of N-acetyl muramic acid and contains L-amino acids instead of D-amino acids. So, that is why the cell wall in these bacteria is a pseudopeptidoglycan, and the plasma membrane in these organisms, the lipids are linked with ether bonds, but in the normal bacteria the linkages are of ester bonds. They are aerobic, facultative anaerobic and strictly anaerobic, that means obligate anaerobic. Reproduction here in these organisms is by means of budding, fission and fragmentation. Similar to that of bacteria, there is no meiosis and mitosis, and the important function of these archaeas, they are helpful in the biogeochemical cycles or carbon, nitrogen and sulfur. In this archaea group, the photosynthesis does not occur, but many of the organisms are phototrophs, that means they utilize the sunlight as a source of energy. Cell structure of archaea, they can stain either gram positive or gram negative. Cell wall, if it is a gram positive, that appears like a single thick homogeneous layer as in gram positive bacteria. That means the gram-positive bacteria contains a thick layer of a peptidoglycan. Like to that of bacteria, the gram-positive archaea also contains a thick layer, and this is a pseudomurine. The pseudomurine, it differs from the true peptidoglycan of the bacteria. How it differs from true murine means it possesses N-acetyl talosaminouronic acid instead of N-acetyl muramic acid. The cell wall in the bacteria is made up of with a peptidoglycan. The other name for the peptidoglycan is a murine layer. It contains the two alternating units of NAG and NAM, that are N-acetyl glucosamine and N-acetyl muramic acid. So, here instead of N-acetyl muramic acid, the cell walls contain N-acetyl talosaminouronic acid here, and there is a tetrapeptide chain in the bacterial cell wall which are majorly of D-amino acids. So, this archaea cell walls contain L-amino acids in its cross links instead of D-amino acids. That makes the pseudomurine but not a murine layer. Examples like Methanosarcina and Halococcus, they have complex polysaccharides which is similar to that of the chondroitin sulfate of the animal connective tissue. Gram-negative Archaebacteria, they lack the outer membrane. Instead, they have a surface layer of protein or glycoprotein outside the plasma membrane. This layer may be of 20 to 40 nanometers in thickness. Now, the plasma membrane in the archaea, it is quite distinctive. In the presence of lipids, they differ from bacteria and eukaryotes having the branched chain hydrocarbons. These branched chain hydrocarbons, they are attached to the glycerol by means of ether linkages rather than the fatty acids joined by ester linkages. In the picture, here it is shown, this is the ether linkages, this is the glycerol moiety. Hydrocarbons, the branched chain hydrocarbons, they are attached to this glycerol moiety by means of ether linkages. Whereas, in the normal bacteria, the cell membranes in the normal bacteria, where the fatty acids, they possess this linkages, they are COO, that is ester linkages. CO is an ether linkage, here COO is an ester linkage. These ester linkages are present in the lipids of the cell membranes of normal bacteria, true bacteria. Bacterial plasma membranes, they contain glycerol diphytenyl diethers and diglycerol diphytenyl tetraethers. Diethers are generally, they are bilayer membrane and contains 20 carbons in length, whereas tetraether, they are 40 carbons in length and they are much more rigid monolayer membrane. So, this is the appearance, the phospholipid monolayer in the archaea and phospholipid bilayer in the bacteria and eukarya. In the eukaryotes and the bacteria, the cell membrane is a bilayered lipoprotein layer. Here in this archaea, the phospholipid monolayer is present. The tetraether side chains, which are of 40 carbons in length, are mainly present in the membranes of thermoacidophilic Archaebacteria. The plasma membrane also contains the polar lipids such as phospholipids, sulfolipids, glycolipids, etc. Moving to the genome in this archaea, the genome is a single, circular, closed DNA contains the guanine and cytosine content that ranges from 21 to 68%, and the genome here it is smaller than the Eubacterium. Some of the differences that show with the bacterial genome on the enzymes are the genome is smaller than the Eubacteria, and here it contains a single DNA polymerase for the replication of DNA, whereas the bacteria possess DNA polymerase 1, 2 and DNA polymerase 3, which have different functions in the replication of DNA. And Archaebacteria, they have a complex RNA polymerase. RNA polymerase, here it is a DNA dependent RNA polymerase. That means this enzyme synthesizes RNA utilizing the DNA template. This DNA dependent RNA polymerase is complex in Archaebacteria and it is resistant to the antibiotic rifampicin. mRNA, that is much smaller than that of Eubacteria, and this bacteria here the DNA contains the introns, where the introns are not generally observed in the bacterial genome. And the tRNA lacks thymine. Instead of that, it contains 1-methyl pseudoiridine. In the tRNA has loops like anticodon loop, DHU loop, T-Cy loop and amino acid attachment site. So, tRNA lacks thymine, but instead it contains 1-methyl pseudoiridine. Like to that of bacteria, they contain the 7-days ribosomes, but the 7-days ribosomes, the shape is variable and it differs from bacterial ribosomes in being resistant to the chloramphenicol and kanamycin drugs. Coming to the physiology and metabolism, they are diverse physiologically and ecologically. They are mostly anaerobic and few are aerobic organisms and facultative anaerobic organisms. Majority of these archaea are either thermophilic or hyperthermophilic, and a few are also in mesophilic. That means thermophilic means in high temperatures, hyperthermophilic means they can live in the very high temperatures, that means above the 100 degree centigrade, and mesophilic, that is the normal temperature that ranges in between 30 to 45 degrees centigrade. Nutritionally, they range from chemolithoautotrophs to organotrophs. Very few members, they carry out an unusual form of photosynthesis. Photoautotrophy is the feature observed in the plants, algae and a few bacteria. The usual photosynthetic process utilizes the chlorophyll, but in the archaea unusual photosynthesis is observed. That means they fix the carbon dioxide, but the fixation of carbon dioxide involves the reductive acetyl coenzyme A pathway or the HB. That means 3-hydroxypropionate. HB means hydroxypropionate. HB, hydroxybutyrate, and DC means dicarboxylate. HB means 4-hydroxybutyrate. So, in the archaea, the 3 different carbon fixation pathways are observed. One is a reductive acetyl coenzyme A pathway. The second one is a HB pathway that includes the 3-hydroxypropionate 4-hydroxybutyrate pathway and dicarboxylate or 4-hydroxybutyrate pathway. Among these, the reductive acetyl-CoA pathway is most energetically favorable. Few members like sulfolobus and thermoprotease, they incorporate the carbon dioxide by means of a reductive CCA cycle. Reductive acetyl-CoA pathway is most energetically favorable for the organisms. Here, in this pathway, 2 carbon dioxide molecules are reduced and incorporated into a acetyl coenzyme A. This is one CO2 and the another CO2. These two are incorporated in this CH3COSCOA, that is acetyl coenzyme A. So, 2 carbon dioxide molecules are reduced and is incorporated into acetyl coenzyme A. The first carbon dioxide, that is reduced to carbon monoxide by an enzyme carbon monoxide dehydrogenase. This also has a function like acetyl coenzyme A synthase. The second molecule of carbon dioxide is reduced to the level of CH3. This X represents the tetrahydropterin coenzyme, to which this reduced form of carbon dioxide that will be bound. Methanogens use this pathway and form this acetyl coenzyme A and produce methane. Some other anaerobic bacteria like planktomycetes, proteobacteria and spirochetes also use this reductive acetyl coenzyme A pathway to fix the carbon dioxide. Another one, that is HB. HB means hydroxypropionate. HB means hydroxybutyrate. 3-hydroxypropionate or 4-hydroxybutyrate is a cycle. HB cycle, it is found in the members of the Crinoarchaeae in the order sulfolobales. In this archaea, 3 carbohydrate catabolic pathways have been identified, that is modified EMP and 2 variations of Entner-Deodorff pathway. 3 carbohydrate catabolic pathways have been identified. One is a modified EMP pathway and the 2 variations of Entner-Deodorff pathway. The modified EMP used by hyperthermophilic euryarchaeotes, which contain the novel enzymes like ADP dependent glucokinase and phosphofructokinase. EMP pathway is also called as a glycolytic pathway, where the initial steps are catalyzed by the hexokinase and it is isomerized and again it is phosphorylated by the enzyme phosphofructokinase. They utilize the ATP, but here in this pathway these enzymes glucokinase and phosphofructokinase, they use the ADP. Unlike in the classical EMP, it leads to the production of AMP. These kinases that results the production of AMP. Another unique step in this modified EMP is the conversion of glyceraldehyde 3-phosphate into 3-phosphoglycerate. In the usual glycolysis process, the glyceraldehyde 3-phosphate is oxidized to 3-phosphoglycerate with the use of NAD as an electron acceptor. But here the glyceraldehyde 3-phosphate is converted directly into 3-phosphoglycerate using the ferredoxin as the electron acceptor rather than the NAD+. The 2 variations of ED pathway are the phosphorylative ED pathway that is observed in the halophiles and non-phosphorylative ED pathway observed in the sulfolobus. This is a ED pathway. In the ED pathway, where the glucose is phosphorylated to glucose 6-phosphate and it is oxidized to 6-phosphogluconate and produce an intermediate compound that is a KDPG, that is a 2-keto-3-deoxy-6-phosphogluconate. Finally, where it splits into one molecule of pyruvate and glyceraldehyde 3-phosphate by an enzyme KDPG aldolase, and finally the glyceraldehyde 3-phosphate it produces a one molecule of pyruvate. So this is the general pathway, modified form of ED pathway, where it has either phosphorylative and non-phosphorylative. The glucose here it is oxidized to gluconate and it is dehydrated to KDG, that is 2-keto-3-deoxy-gluconate, and the KDG is phosphorylated at the expense of ATP to KDPG, and the remaining pathway is similar to that of ED pathway that is observed in the bacteria. So here this is what we call as a phosphorylative pathway, where it involves the ATP dependent phosphorylation of KDG into KDPG. The non-phosphorylative pathway that is used by sulfolobus species, the KDG is cleaved by KDG aldolase to pyruvate and glyceraldehyde. Glyceraldehyde is oxidized to glycerate and this glycerate is phosphorylated to 2-phosphoglycerate. This enters into the glycolytic pathway and produce pyruvate. So in this there is no involvement of ATP here, so there is no net ATP is produced. This phosphorylative and non-phosphorylative variations of ED pathway are observed in the Archaebacteria. Comparison with the true bacteria, Archae, as mentioned, they are the primitive prokaryotes, and due to the presence of distinct characters they are separated. They have a separate domain, that is Archae, and whereas eubacteria they are single celled primitive organisms. They too are present in various habitats and they too have a diversity in the shape and structure. The Archaebacteria are also called as extremophiles because they are found in the extreme environments like deep sea, hot springs, etc. But the normal bacteria, they are present in different habitats. They are either terrestrial, marine, and inside the living and non-living organisms. The archaeal cell wall is made up of with a pseudopeptidoglycan or pseudomurine. That means it contains the L amino acid instead of D amino acid, and N-acetyl talosaminouronic acid instead of N-acetyl muramic acid. Here the bacterial cell wall is made up of with a peptidoglycan which contains N-acetyl muramic acid and D amino acid, and the fatty acids of lipids are linked to the glycerol by means of ether bonds, but here the fatty acids in the membrane lipids of bacteria are linked to the glycerol by means of ester bonds. Archaea generally they do not use glycolysis or Krebs cycle for glucose oxidation. They follow the modified forms of a glycolytic pathway, whereas the glycolysis and Krebs cycle are the important metabolic pathway observed in the bacteria for the glucose oxidation. Another comparisons are the Archaea, though they perform the photosynthesis but they do not release oxygen. Eubacteria, they contain the photosynthetic pigments that can perform the photosynthesis and prepare the starch. The Archaeflagella, they are called as Archaeella, and here they are synthesized by adding the subunits. the flagellin subunits, flagella is made up of with a flagellin. So, the subunits here they are added at the base, whereas in the bacterial flagella they are assembled at the tip of the flagella. Archaea reproduce by fission, budding and fragmentation like to that of bacteria, but they do not produce any spore like structures in the starving period under the unfavorable conditions. But here the bacteria are capable of such a hot resistant structures which can survive in the extreme conditions of the environment. Another features, another comparisons are the thymine that is not present in the tRNA, but it is present in the bacteria. Introns, which are the non-coding regions of the DNA, they are present in the chromosomes of archaea. This character is similar to that of the eukaryotic feature. Introns are absent in the chromosomes of the bacteria. And as explained, RNA polymerase, that is a DNA dependent RNA polymerase in the archaea is complex. It contains more than 8 polypeptides, whereas in the bacteria that DNA dependent RNA polymerase contains 4 polypeptides, and archaea are non-pathogenic. Bacteria, they are either pathogenic or non-pathogenic. And the classification, the second edition of Burgess manual on the basis of 16S rRNA, they divided the archaea into 2 major phylum, that is Crinoarchaea, Euryarchaea, and Conarchaea is also there, but it is not recognized taxonomically. Phylum Crinoarchaea, it consists of organisms that live in extreme high temperatures and are dependent on the sulfur. The sulfur may be used as an electron acceptor in an anaerobic respiration or as an electron donor by lithotrophic organisms. These Crinoarchaea, they are often found in the heated water or the soils that contain the elemental sulfur. Examples hyperthermophilic organisms are Sulfolobus, Pyrolobus. Pyrolobus fumeri, that was isolated from hydrothermal vent of Pacific Ocean, which can grow best at 105 degree centigrade and even 5 autoclaving temperatures, that is 121 degree centigrade for about 1 hour. The 2 best studied members of Crinoarchaea are Sulfolobus, another one is a Thermoproteus. Members of Sulfolobus are aerobic, spherical, and they require the temperature optimum 80 degrees centigrade and pH is a 2 to 3. That is why they are called as a thermoesrophilic organisms. The cell walls in this group, they contain the carbohydrate and archaea specific lipoproteins. These Sulfolobus species organisms, they use H2S or hydrogen sulfide or iron sulfide as an electron donors and oxygen as an electron acceptor. It uses non-phosphorylative version of the AD pathway and a complete TCA cycle. Thermoproteus species, they are long, thin rods and they are present either in branched or bent form, and the cell walls of Thermoproteus contains glycoprotein. Thermoproteus species are anaerobic and they grow at the temperatures at 75 to 100 degree centigrade with the optimum pH around 3 to 4. They are found in the hot springs and hot aquatic habitats that are rich in sulfur. The second phylum is a Euryarchaea. It is a very diverse phylum that contains important groups like methanogens, halophilic methanogens and sulfate reduces even. Methanogenesis process, that means the production of biological methanogenesis process. The biological production of methane was discovered by Volta, and he demonstrated the importance of the natural gas in the industrial purpose. A few points about methanogens are the largest group of cultured Archaebacteria. It contains 5 orders like methanobacteriales, methanococales, methanomicrobiales, methanosarcinales and methanopyrales. They are autotrophic and they use the reductive acetyl coenzyme A pathway to incorporate the carbon dioxide. Methanogens are the obligate anaerobic organisms. They obtain the energy by oxidizing hydrogen or the formate, and they grow as an autotrophic organisms. They live in the anoxic environments. They are obligate anaerobic organisms. They live in the anoxic environments and produce a match gas. The methanogens, they contain the unique coenzymes. They are coenzyme 420 and coenzyme M. The cell walls of methanogens, they contain the pseudomurine. That means they contain the N-acetyl talosaminourenic acid instead of NAM, and tetrapeptide chain with the L amino acids. And the habitat for these methanogens include, they are the marshes, swamps, ponds and the ruminants of an animals, and then the anaerobic sludge digesters. The important cofactors are the methanophuran and methanopterin, coenzyme 420 and coenzyme M, F430. At the end, the unique features of Archaebacteria to be remembered are they possess the cell membrane with a unique chemistry. That means the ether linked phospholipids are present, and their genes, they contain the intron, the feature of eukaryotes, that is what unique gene transcription, and the single chromosome like that of bacteria, but the gene transcription process is similar to that of the eukaryotic cells. Archaebacteria are capable of methanogenesis process. Is the process of production of methane gas, which is a form of anaerobic respiration process that release the methane. And the differences in the ribosomal RNA that suggest that they have diverged from bacteria and the eukarya. So that's the reason they possess the features of both the bacteria and eukarya. Thank you.